A cell is an incredibly complex structure with a huge variety of biomolecules that are densely packed in all kinds of sophisticated arrangements. For example, here you see microtubules, which are formed by tiny little proteins that we imaged with a fluorescence microscope. These proteins form the cytoskeleton of the cell, and they are crucial in all kinds of cellular processes. Now, cells and the biomolecules they contain can be incredibly small, down to a millionth of a millimeter, which is why we use microscopes to visualize them. However, the problem with conventional optical microscopes, such as this one, is that they have a resolution limit which is about 200 times larger than the size of the smallest biomolecules. In other words, if we were to zoom in on this cell, we wouldn't observe more detail, but instead the image would simply become blurry. In my MSCA fellowship project, I am breaking this resolution barrier by measuring very precisely the position of individual molecules in the sample. To do so, we built a custom laser scanning microscope in our lab. The idea is to scan a donut-shaped laser beam around a single molecule of interest in the sample. We then measure the response of the molecule to the laser light with an extremely fast and sensitive detector called a spot array. This will allow us to localize the molecule with a precision of a millionth of a millimeter. Once we know where the molecule is, we can move to the next one, and so on. We repeat this process until we have found the position of hundreds of molecules. Then, in post-processing, we can use this information to build an image. Similar techniques have already been developed recently, but now, thanks to the spot camera, a lot of improvements can be expected, including the possibility to visualize cells with molecular resolution, which will allow us to understand with unprecedented detail how cells work.